Welcome to topic 9 of marine aquatic ecology, and this topic continues with competition. We're going to look at the actual equations, the Lotka-Volterra equations, differential equations a little bit, and then we're going to consider some classic examples of competition, studies of competition in nature and the marine environment. Then we move on to the consumer resource interaction, predator-prey interactions, and the isocline analysis of those in population density phase space. And in this case, in this type of interaction, we often see oscillations. They can be wide oscillations, and they can also cause exclusion of the prey species, sometimes the predator species as well. So we're still at the population level of analysis, and we're still talking about interactions. All right, now we will actually consider the equations, the differential equations that were used by Lotka and Volterra starting in the 1920s. Al Lotka and Vito Volterra both published essentially the same paper without knowing the other one did it in the 1920s, describing these equations. So let's take a look at it. So these, these equations were used to describe exploitative competition dynamics and also predation, and were used to create the, the graphical isocline analysis. So these are just simple changes to the standard logistic growth equation that take into account the competitor's ability to reduce the maximum population size possible, or the realized carrying capacity. So these equations basically inaugurated the field of population ecology. So it really started in the 1920s. And in the 100 years since then, more and more realistic treatments have been used to predict dynamics of populations of competitors, predators, mutualists, diseases, and hosts. And those same equations are used to predict the spread of the coronavirus right now. And other diseases. So they're also the basis of the field of epidemiology. But it all started with Lotka and Volterra, who happened to come up with the equations for exploitative competition independently in the 1920s. So the logistic growth equation is visualized in a plot of R against N. So, and this is the logistic growth equation. Remember, it's a sigmoid curve where the population increases to carrying capacity. And it's caused by this plot of R against N. As N increases to carrying capacity, little r decreases along a straight line from R0 at its maximum when the population is small to 0 at its minimum when N is equal to K. So that's just one population. And when you add individuals of another species in competition, then these individuals reduce R even more. So this is intraspecific competition here. We're only looking at one species. Within this one species, as you increase its N, competition among individuals within that one species causes little r to drop until it's 0 at the carrying capacity. So the population no longer increases at the carrying capacities. That's intraspecific competition. If there were no interspecific competition, in other words, resources are infinite, then little r would stay constant. And what kind of growth is there when little r is constant? That's exponential growth, or geometric growth. Constant little r, that's geometric growth. But in reality, this will happen, and eventually population per capita rate of increase drops down to 0 at the carrying capacity. Right. What happens when you add another population of competitors in? Well, you have interspecific competition reducing this entire curve even more, depending on how many individuals of the competitor are there. So the entire curve drops down to some lower level, depending on how many individuals are there, and this is caused by both intra and interspecific competition. So both are going on at the same time when we have this curve here. So intraspecific competition is just the logistic growth equation for one population. Interspecific competition is a logistic growth equation for two populations, one equation for each population. As n increases, the entire line for R decreases in the other species. Okay, so this line is the equation right here for little r that we already talked about. And this line is the equation for little r for both species. So this is just your own species right here, reducing little r. But now we have the other species in here too in this equation, in this term right here. It's competition coefficient multiplied by the number of individuals of the competitor. So when you add N1 plus A12 times N2 together, you get the total number of equivalent individuals of species 1 in terms of their consumption rate of species 1's limiting resource. When you add these two together, it brings species 1 closer to its maximum population density or realized carrying capacity. And when that maximum is reached, the rate of increase of species 1 drops to 0. So this is how the density of one species affects the fitness of the other species through its effect on little r of the other species. So the entire line relating r to n drops in the presence of the competitor. The amount by which it drops is determined by the competition coefficient. The higher the coefficient, the stronger the competitor, and the farther r drops, little r drops, in the presence of the competitor. Okay, so, so here are the two equations for the competition situation. So we've got two species competing, and each one lowers the other one's growth rate, per capita growth rate, little r. Through this term here, population density multiplied by the effect, the competition coefficient of that species. Species n multiplied by its competition coefficient converts this number into an equivalent number of species 1 in terms of the effect on its limiting resource. So we just add this term into both to get Lotkin-Volterra's competition equations. Okay, that's basically it. And this competition coefficient just converts species 2 to species 1. So for example, if one individual of 2 equals 2 ones, then A12 is equal to 2. And that's how you convert from 2 to 1. So you have the same units when you add them together. That just means that species 2 consumes twice as much resource of species 1's limiting resource than 1 does. So its competition coefficient is 2. So that's what you multiply to get an equivalent number of individuals of species 1. All right, so this is the logistic growth equation here. Little r is equal to this. And this is the Lotka-Volterra competition equation for the logistic growth equation in the presence of the competitor. Just add this term on to get the effect of the competitor on the growth rate and the fitness of the other species. Now we can visualize that in this diagram. So the density of one species affects little r of the other species. And that effect is expressed through the competition coefficient. Okay. And there's the effect right there. So the density of this species, N2, affects the fitness, R1, of this species through its competition coefficient, how much resource it consumes of species 1. And the same vice versa. Once we have these equations, we can set them equal to 0 and solve for n to get the zero growth isoclines. That's all we do. Set them to 0, that means zero growth, and then we get the equation for the line of zero growth. So you set this little r to 0, or the growth rate to 0, and we get this equation for the zero growth isocline. This is the zero growth isocline for species 1. So there it is. This is the line, and this is the equation for that line in terms of the two ends, and then these two parameters of the line. So this gives the values of N1 and N2 for which the growth rate is 0. So any values of N1 and N2 along this line give a growth rate of 0 for species 1, the yellow species. And we can do the exact same thing for the other isocline. This is the equation for the line. All these values of N1 and N2 give a growth rate of 0. So this is a zero growth isocline for species 2. All right, so now it's trivial to derive the shutout level from these equations. Okay, so for this red line, we just say N2 is 0. And then we solve for N1. So 0 equals K2 minus A21 times N1. Then we solve for N1. Well, N1 is equal to K2 divided by A21, which is the shutout level here. And what's the shutout level of species 2? Well, we just set N1 equal to 0. So the shutout level of species 2 is just K1 over A12, which is this right here. So that's the algebra. Those are the equations that give us these shutout levels. So we figured them out sort of intuitively last lecture, and now we just show them mathematically using the equations. Okay, so the Laca-Volterra approach gives us several simple insights here. And these are that, number one, a limiting resource is a resource that if you reduce it, it reduces fitness. A competition coefficient for species 1 is the number of individual species 2 equal to one individual species 1 in terms of limiting limiting resource of species 2, effective fitness on species 2. The shutout level of species 1 is the number of individuals of species 1 equal to K2 individuals of species 2. And the zero growth isocline of the first species, is all combinations of the numbers of the two species in the populations that cause zero growth of species one. All right, so that's simple mathematical theory, but does it accurately describe nature? Well, all models are technically wrong, but some are actually useful and describe processes closely enough to be predictive. So let's look at some real species competing in the laboratory for resources that we can monitor and then ask exactly what happens to limiting resource levels as competing populations increase their populations, their numbers alone, and as they increase together. And what happens is they shut each other out to the populations and the resources. Right, consider two diatoms here, Asterianella and Cynedra, which are freshwater phytoplankton, Lake Michigan, the USA. And they consume the limiting resource silicate, and were studied by Tillman in 1981, paper in Ecology. These can be grown in the lab with a constant supply rate of silicate, and we can monitor both populations, and we can monitor the silicate. So over time, each increases following a logistic growth curve, and each one brings the silicate down to a level in their environment, down to the minimum value at which the rate of consumption equals the rate of supply. So here the rate of supply of silicate is equal to the rate of consumption in both of these. So the populations increase to the level at which the silicon supply rate can support it. So as you can see, each species reduces the level of silicate by a different amount at equilibrium, at carrying capacity. So Cynedra alone can reduce the silicate to a lower level at equilibrium and can still maintain its population at a lower level. As you can see here. When the two species are grown together on the same supply of silicate, the Cynedra wins in competition because it can subsist on a level of silicate that is below what Asterianella can subsist on at its carrying capacity. So if the silicate drops to a level below the minimum that Asterianella can subsist on, then the Asterianella will drop from zero population growth to negative growth and eventually reach a population size of zero. It will be shut out. So what's the population density that Cynedra needs to reach in order to do that? Well, if it reaches this density right here, then it brings the silicate level down to a level where Asterianella's growth rate is no longer positive. And so this is the shutout level of Cynedra. If it reaches this level, then it shuts out the Asterianella. All right, so growing together, we can see again that this is the shutout level of Cynedra, reaches this density, and the silicate level is low enough that Asterianella then starts to be negative growth. So this is competitive exclusion, the outcome of the struggle for existence while supported by a single limiting resource, silicate. So we can predict the outcome of competition just by comparing the resource levels of two species in isolation. So if one species can reduce the resource to a level below the minimum level that the other species can subsist on, then it wins or can win the competition. So just by comparing the resource curves to the population curves, we can determine the shutout levels and carrying capacities and determine whether the shutout level is greater than the carrying capacity or not. So the winner is a species that reduces the level of limiting resource the farthest. All right, how does this result conform to the Lotka-Volterra theory? Well, the LV theory does not predict what the ambient level of limiting resource should be at any given time. So it does not predict that one species will bring that level to a lower value than the other. What the LV theory predicts, the Lotka-Volterra theory, is that both species on the same limiting resource will reach their shutout levels and carrying capacities at the same time. Therefore, either species can win. But in this example, Cynedra always wins because it reduces the level of the resource, the silicate, to a lower value and is still able to survive and reproduce, while the other species cannot at that low level of resource. So, what is wrong with the Lotka-Volterra model? Well, in cases where models don't seem to work, they can help us understand why they do work in other cases. So, the Lotka-Volterra equations make an important assumption that may not be met in nature sometimes. Consumption rates of limiting resources are always constant. If they are always constant, then we would not see the results with Cynedra and Asterianella, with the predictable winner every time on the same resource. What appears to be happening with these two diatoms is that when resources reach low levels, some species might not be able to maintain the same consumption rate because the resources are harder to find or take longer to reach or are taken up by cells more slowly. So if one species has a lower rate of resource capture when resources become rare, then that is another reason that it can be competitively excluded. That is not revealed in the Lotka-Volterra equations. It assumes a constant rate of consumption. So Cynedra appears to win when grown with Asterianella because it is capable of consuming silicate faster than Asterianella at low levels. And so it brings it down to even lower levels that Asterianella cannot subsist on. And this might be a phenomenon that occurs commonly in competing species. So one way to make the Lotka-Volterra equations more realistic is to make consumption rates dependent on the resource levels. So this gives us a more realistic view of shutout levels. It can also be determined by how efficiently a competitor can consume resources at low resource levels. So by looking at the resource curves for the two competitors, these resource curves, we can see immediately what those shutout levels are and what the carrying capacities are, and make predictions that are often useful, as in the case of Asterianella and Cynedra. One important outcome of either kind of analysis, whether you're looking at the Lotka-Volterra competition equations or laboratory resource consumption under the assumption of a single limiting resource, one outcome is called the competitive exclusion principle. So this principle states that two species that are dependent on just one single limiting resource cannot coexist. Or equivalently, for two species to coexist, they must be limited by two or more different resources, allowing one or both to switch to different resources when in competition. This principle is also sometimes stated as one niche, one species, with the idea that two species cannot occupy the exact same niche indefinitely. One will eventually displace the other, the superior competitor. So this seems intuitively convincing. For any two competitors, they can't be exactly equally competitive. One species will always be at least a little bit superior. So eventually, that species should be able to win out. over the other. So one species will have the higher little r, 0. It'll be better able to capture resources when scarce, and so on. So if their complete competitors cannot switch to different resources, but are both dependent on the same identical resource, then one will always be superior, and we can't expect them to coexist indefinitely. So if you extend this to n species, for n species to coexist, they must be limited by n different resources at least. This principle also, of course, assumes that it's possible to reach equilibrium. In reality, equilibrium may be rare or difficult because there may be regular disturbances in the environment. For example, density dependent or independent disturbances that prevent a competitor from reaching its shutout level. Predators might prevent this from happening, or the winter season might prevent it. And if so, then some kind of de facto coexistence is possible, even though one species is a superior competitor. But if equilibrium eventually occurs, then coexistence, according to the Lotka-Volterra equations and lab experiments, coexistence requires a different limiting resource for each competing species. All right, so now we can see a bit more clearly how coexistence can occur or not occur by considering the same population resource graphs as we did for Cynedra and Asterianella, but now we allow the species to have two different limiting resources. So no matter what the limiting resource is, the species who can reduce the level of the competitor's limiting resource to a level so low that the competitor has difficulty capturing it, that's the species that can reasonably be predicted to be the winner of, the struggle for existence. So here's a case where coexistence is predicted. So in this case, both species brings its own limiting resource down to a lower level than it brings the other ones down. So it's more limiting to itself than it is to the other species. So in this case, the carrying capacity of both is lower than its shutout level. So neither species can exclude the other one. So this is a case of coexistence. And it requires two limiting resources. So this is case 4 in the Lotka-Volterra models. And to achieve it, we have to have two limiting resources. So here's a case where either can win because both limits the other species more than it limits itself, because both species shutout level is below its carrying capacity. So it can easily reach its shutout level before it reaches carrying capacity and exclude the other species. All right, so this is case 3 in the Lotka-Volterra model. Two limiting resources. Seems like this situation is probably not likely in nature, because it just seems unlikely in principle that a species would be better adapted to consuming another species' limiting resource than it would be in consuming its own. But, it's a theoretical case here. It's just possible theoretically. So this is case 3. Either species can win the competition. And here's a case where species 1 wins, and the symmetrical case where species 2 wins in terms of two limiting resources. So we have some fairly simple rules, whether we look at it from the lab experiments or whether we look at it from the theory. So if both species have two competitors and they both depend on the same limiting resource, then one will always win eventually, unless something is preventing it from winning. And if each depends on a different limiting resource, then there are three possibilities. One can still win, both can win depending on the starting conditions, and there's a possibility for coexistence. So, coexistence only is possible in theory if there's more than one limiting resource limiting the two species. So we can interpret competitive exclusion or coexistence in basically two complementary ways. We can think of it in terms of efficiency of resource consumption at low resource levels, as suggested by lab experiments. And second, we can assume constant rates of consumption relative to carrying capacities, which is a situation that the Lycka-Volterra equations look at. All right, so let's return to our initial example of Gauss and the two paramecium species grown in the lab in competition. He found that the species Caudatum and Berseria, when grown together, always were able to coexist, but Caudatum and Aurelia, when grown together, could not coexist, never coexisted, and Caudatum always won the competition and excluded the other. All right, with further experimentation, he found that in the coexistence example, the Caudatum and Berseria, the two species were limited by two different resources. Caudatum was subsisting on bacteria, while Berseria was subsisting on yeast. So with two limiting resources, we have coexistence. All right, so we saw a classic example of competition in the lab, Gauss's experiments. Let's now look at field studies of competition, more recent. So we'll take a look at some classic studies of competition in nature that were elucidated in field experiments. And probably the most famous and often cited study of competition in the field is a marine example. That's Joe Connell's 1961 study of competition for space between two species of barnacles on the Scottish Rocky Intertidal. And of course, the Rocky Intertidal in general is a habitat that is kind of a natural laboratory for field experimentation and ecology. You can easily manipulate the population density of sessile species like barnacles, mussels attached to rock, and monitor the effect that manipulation has on other species and proxies for fitness of other interacting species, such as competitors. So, to test the hypothesis that species 1 competes against species 2, you can remove species 1 and see if species 2 responds positively, and vice versa. Removal of barnacles, of course, is easy. You just scrape them off the rock with a chisel, which is what Connell did for his PhD thesis research. He just went out and chiseled barnacles off the rock and then took photos to monitor what the response was of neighboring competitive barnacles. It's a bit more difficult to add individuals to increase N, which is why most experiments of the Rocky Intertidal are removal experiments and not addition experiments. You just go and just chisel off species. So in this study, Connell looked at two species of barnacles. These were Thamylus and Balanus that grow in the upper intertidal in Scotland and many places throughout temperate latitudes. And he observed that adult barnacles of the two species were distributed differently on the shore. Balanus tended to occur lower in elevation on the shoreline on the rocks, while Thamylus occurred at higher elevation. And adults of the two species had very little spatial overlap, if any. However, larval and juvenile distributions did overlap quite a bit. So barnacles have a pelagic larval phase, and they settle on the rock over a larger elevation span than the adults. So the adults of Thamylus higher up than the adults of Balanus. Larvae settled out over a wider range and did have quite a bit of overlap. So the lower portion of the Thamylus distribution of larvae and juveniles overlapped substantially with the upper portion of the Balanus distribution. Connell wanted to know why these differences in distribution occurred. Could it be that the two species just occupied different fundamental niches on the shore, or could competition be part of the explanation? Could they have similar fundamental niches but different realized niches because of competition? Why did the adult distributions have very little overlap? To answer that question, he scraped Balanus and or Thamylus adults right off the rocks at several locations that differ in tidal elevation, and monitored over several months how the other species, the other barnacle species, responded to the manipulation. So compared to controls, which were not scraped. And he found that where he scraped Balanus off the rocks in lower areas, the Thamylus adults increased in abundance, spreading down in tidal elevation well into the zone that was formerly occupied by the Balanus. All right, so if you look at this graph, this is the number of Thamylus species. So both the bold and the dotted line are the number of Thamylus species individuals, the density of the Thamylus at these different months and years, 1954 and 1955. And you look at where he removed the Balanus, the Thamylus individual stayed relatively constant over the time. Whereas areas where he did not remove the Balanus, the Thamylus population went way down, was excluded. And these little numbers here, plus 3.5 feet, that's the elevation on the shore. So high on the shore, there was not much of an effect of removal on a Thamylus population. But if you go low on the shore, this is close to the zero tide level, there was a large effect of Balanus removal. All right, here's another diagram that is a little bit simpler. Okay, so here we have the adults and larvae of the Balanus, the adults and larvae of the Thamylus. Notice the larvae overlap quite a bit. High on the shore, there's not much difference between the dotted and the solid line. It shows that there was not much of an effect of Balanus removal. Go down in this region where there's a larva overlap, there's a big difference here between areas where the Balanus was removed and where it was not removed. And down here, both decreased, regardless of whether they were removed or not. All right, so where he scraped Balanus off the rocks, Thamylus adults survived, and survived even down in the lower tidal elevation, well into the zone that they don't survive if the Balanus is not scraped off the rocks. And where he scraped the adult Balanus off in higher areas, the Thamylus response was no different from controls. And on the other hand, scraping off Thamylus generally did not result in any effect on Balanus. So this indicated that competition might have excluded the Thamylus from the lower Balanus zone, but competition had no effect on either species in the upper Thamylus zone. Okay, so to understand why Balanus did not occur higher on the shore in the Thamylus zone, up in this region, He moved rocks with attached adult balanus to higher elevations on the shore and monitored their performance. And he found that balanus on these rocks died from desiccation, just dried out during low tides, especially during warm temperatures in the summer. So desiccation was great on the balanus up in these higher levels and is responsible for the fact that you don't see adults at these higher levels in balanus. And the desiccation effect was not visible for Thamylus unless you went even higher on the shore. So Thamylus was better adapted to desiccation than balanus at the higher levels of the shore. So we also wanted to verify that competition was excluding Thamylus from the lower portion of the shore. We want to verify it. So we followed the fate of larvae and juveniles as settled into the lower region, where adult balanus occurred. And here is where he saw what was going on. So as the balanus juveniles grew to larger size, they overgrew or undercut or crushed the Thamylus. All right. So the balanus apparently has stronger shells that Thamylus is unable to defend itself from, at least in the lower intertidal. But the balanus was not able to overgrow or undercut or smother the Thamylus up in the higher zone where Thamylus adults occurred, up in this part of the zone. So balanus was not able to competitively exclude Thamylus in this region. So the main conclusion of Connell's was that competition with balanus was preventing Thamylus from spreading down into the immediately lower intertidal, but competition was not preventing balanus from spreading up into the Thamylus zone. Rather, desiccation was preventing that from happening. So apparently balanus is a superior competitor in the lower zone, but not in the upper zone, because it is not as well adapted as Thamylus to the desiccation in the upper zone, which is exposed to the air more often at low tide. So this study was the first of several studies to suggest a general rule of species distributions in the rocky intertidal, and that was that the reasons that any species does not occur lower in the intertidal was generally for biological reasons, usually competition and often predation kept it out of the lower intertidal. But the reason it doesn't grow into farther into the upper intertidal than you see is because of non-biological factors, physical factors, usually desiccation or heat stress are the major factors keeping species from going farther into the upper intertidal than you normally see them. So we can interpret the situation in terms of the Lotka-Volterra equations by simply drawing isoclines that are in agreement with the main experimental results. So first, balanus has a carrying capacity equal to the maximum density possible on the rocks in the intertidal zone in the absence of Thamylus. In the lower intertidal, if you add Thamylus individuals, it will have little effect or no effect on the balanus density, indicating that the isocline for balanus, the yellow line here in the figure, is almost vertical. So it's just the same no matter how much Thamylus is there. But in contrast, consider the Thamylus carrying capacity, also the maximum density in the absence of balanus. So if you then add balanus, the density of Thamylus will drop off rapidly due to competition with balanus. So we can see obviously at the shutout level, balanus is essentially its carrying capacity. As if it occupies a shore in the lower levels, it's robust enough that Thamylus cannot invade. In contrast, the shutout level of Thammus is very high, maybe does not even exist, indicating that Balanus wins this struggle easily in the lower intertidal. But in the upper intertidal, neither species can win the competition, suggesting that there's a stable coexistence caused by the fact that Balanus is at the limit of its ability to survive, reproduce, and compete at the upper intertidal levels where Thammus thrives, and Thammus is not able to overgrow Balanus because of its weaker shell. Okay, so this is a coexistence case here. The two isoclines cross at a stable equilibrium here, where both exist together. All right, there are hundreds of similar marine examples of competition experiments in which population density of species are experimentally modified, and researchers monitor the response of other potential competitors. In the rocky intertidal habitat in particular, many manipulative experiments have been performed because it's so easy to do so in this habitat. Most of these were done in the US, both coasts in the UK and Australia. Very few have been done in the Mediterranean. So there's opportunities for people to make a name for themselves by doing this type of experiment in the Mediterranean. Although rocky intertidal isn't quite as impressive here as it is in other places because the intertidal region is not as wide. So we'll talk about some more of these a little bit later. All right, next topic is the evolutionary dynamics of competitors. How do competitors evolve over evolutionary time, over millions of years? Well, for Darwin, competition was a prime trigger for evolutionary change. He envisioned natural selection as ubiquitous in nature, favoring individuals that survived the struggle for existence as a result of evolving some new and better ways to capture scarce limiting resources and therefore become better competitors. And in evolving new ways to become better competitors, Darwin believed that new species often evolved. This was an important enough mechanism for how species originated that it became the title of his book, Origin of Species. All right, so here's his basic argument as we would express it today with some important new terminology. So imagine we have two competitors and one is superior to two. So it's very likely that these species have two different distributions in nature. They don't occur together everywhere. Where they occur together in the same geographical region or area, they're living in what's called sympatry, together in the same place. And where they occur separately in different geographical regions, they're living in allopatry, different places. Okay, so where these two species are living in sympatry, they may often be living in direct competition with each other, either exploitative or interference or both. So if they're in direct competition, then the superior species, species one, will tend to exclude the inferior species two. This means that for species two, the inferior competitor, the population death rate will exceed the birth rate here in the area of sympatry. Individuals may migrate into the areas of sympatry regularly, but they will not be able to maintain populations there without migrants because the death rate is greater than the birth rate. So areas of sympatry can be considered sinks, sink areas, while areas of allopatry can be considered source areas of migrants from the species two. The migrants help to make up for the lower birth rate. In areas of sympatry, we predict that there should be strong selection on migrants of species two that avoid mortality due to competition. So individual variants able to escape competitive death should be favored. If an inferior competitor remains in sympatry with the superior competitor over a long period of time, over evolutionary time, maybe thousands or millions of years, then the inferior competitor has the opportunity to evolve in response to competition. The superior competitor also is expected to evolve in response to competition if competition lowers its fitness somewhat, even though it eventually wins the competition. Since competition is a negative-negative interaction, we would expect species in general to evolve to reduce the effects of that competition, to reduce the negative effects on your fitness. And how competitors evolve to reduce the negative effects of competition on their fitness, and those living in sympatry over long periods of evolutionary time, how do they do that? Well, one way is called character displacement. In other words, traits that are related to consumption of limiting resource evolve to be different between the two competing species while they are in sympatry. So, character displacement is just the observation that the difference between two competitors in some trait is greater in sympatry than in allopatry. So, character displacement is consistent with the hypothesis that in sympatry, competitors who evolved the ability to switch somewhat to a different resource or a different part of the same resource will be favored by natural selection and will allow them more success in the struggle for existence. Species whose resource consumption traits have evolved to be somewhat different in sympatry are likely to rely somewhat differently on those resources and will likely experience less competition as a result. So, Joe Connell called this character displacement the ghost of competition past. Uh, it's just an interesting reference to the Christmas Carol story of Ebenezer Scrooge, who had to endure the ghost of Christmas past. Okay, so the idea that competition occurred in the past caused evolution of character displacement, which relaxed or avoided competition in the present. Right, Charles Darwin's classic example of character displacement were the finches, the genus Geospiza on Galapagos Island, which he studied as a young man during his travels to study natural history, geology, ethnology, and so on while on the ship, the HMS Beagle. At this time, he was 22 years old and had recently graduated from college. And there are several islands, and each of several finch species occurs on one or more of the islands. So here's a very nice natural experiment where you can statistically compare the bird's competitive traits in sympatry and in allopatry. Sympatry when two species are living on the same island, and allopatry where the same two species are living separately on different islands. So the competitive trait that's most obvious is the trait used for feeding, their beak and its size. And he found that on islands where two species occurred together in sympatry, for these two species of finch, the distribution of beak sizes of the two birds hardly overlapped at all. So the small beaked one was quite small, the large beaked one was quite a bit larger than you'd see on islands where they're in allopatry, where the other species is not present. So where the same two species live separately on different islands, the two beak sizes almost completely overlapped. Completely overlapped. They're almost the same beak size here. So this, he explained, is an observation consistent with his hypothesis about the struggle for existence. If two species are competing for resources, individuals that are better able to consume resources when they're in short supply will be favored by natural selection, which causes evolution. And one way of consuming resources in short supply is shifting to a different resource or a different part of the same resource that is not in such short supply. And finches generally feed on seeds, and their beaks are adapted to break open hard shells to get at the meat inside. So the size of a finch's beak to a large extent, determines the strength that it can use to break open the seed. Larger beaks can feed on a wider range of seeds than smaller beaks, but tend to concentrate on larger seeds because they have more food value. So his explanation for the greater difference in beak size between sympatric species than allopatric species, he interpreted it as caused over millions of years of competition for the same limiting resource, namely plant seeds of a medium size. Over time, selection favored birds in sympatry that were able to shift to different sized seeds, and those were on average birds that had different sized beaks. So the Darwin's finch example is the classic example of competitive character displacement, the idea that competition can shape the evolution of divergence in consuming ability and divergence in resources consumed if species are in sympatry and competing over millions of years. And ever since Darwin, people have been looking for similar beautiful examples of character displacement. Joe Connell coined the term ghost of competition past. And so lots of people have been looking for this ghost of competition past in their favorite species. So another classic but more recent example of character displacement is an aquatic example. This is a marine freshwater example, estuary example of the three-spined stickleback, Gasterosteus aculeatus, and other species. A small brackish or freshwater fish in the Pacific Northwest. So in this species, the male builds nests for females who lay eggs in the nest while the male fertilizes and guards the eggs from predators and from other sneaker males who resemble females and try to trick the male in order to fertilize the eggs themselves. So these fish feed using gill rakers, which are structures attached to the gill arches that allow fish to filter food particles out of the water as the water passes through the gills. So many fish are suspension feeders that use gill rakers to filter out food particles in the same way that baleen whales use baleen to filter out food particles. Other fish feeding this way include mullet, herring, anchovy, and sharks, such as the megamouth, basking, and whale sharks. And the size and structure of the gill rakers are thought to have evolved to optimize feeding on a particular component of the plankton, particular size of the plankton. So Schluter and McPhail, Dolph Schluter, who's a friend of mine, in the 1990s found character displacement in gill raker size in different populations of three-spined stickleback, very similar to what Darwin found in Darwin's finches. All right, only here, instead of having islands in the water, we have actually islands of water, namely lakes, that are separating the species. So plate tectonic movements have caused the uplift of the coastline of British Columbia, Canada, over the last several millions of years. As a result, many former estuaries have risen above the high tide level and no longer have contact with the ocean. So become freshwater lakes. So now sticklebacks occur both in estuaries and in freshwater lakes. In some lakes there are two species or more, and other lakes there are just one species. So we can look at gill raker morphology in species in allopatry, where one species is found by itself in a lake, and in sympatry, where the same species, two species, are together living in the same lake. What Schluter and McPhail found was exactly the same thing that Darwin found with Darwin's finches in the Galapagos Islands. Instead, we're looking at lake islands. So if you plot the distribution of gill raker length adjusted by size of the fish, you found, just as Darwin did, that the gill raker length distributions almost did not overlap at all between two species in the same lake, while either species alone in different lakes had gill raker distributions that completely overlapped. Not only were there two different non overlapping distributions of gill raker length in the two species, but the two species also lived in separate habitats within the lake, where their gill raker lengths were adapted to the food supply in those habitats. The species with the longer gill rakers fed in the limnetic habitat, that means in the lake water column, while the species with the shorter gill rakers fed in the benthic habitat at the bottom of the lake. So in sympatry, there was complete separation of the two species into different habitats, different food supplies that were exploited with different gill raker morphology. In allopatry, the same species did not forage in different habitats and had the same ranges of gill raker lengths. Again, their observations are consistent with Darwin's predictions of the evolutionary outcomes of the competitive struggle for existence. In the presence of competition, selection favors evolution away from the common resource that the two species are competing for. So this relaxes the intensity of competition so that it does not have as strong a negative effect on fitness as previously. So not only was gill raker length different in the two sympatric species, but the benthic was larger and the coloration different as camouflage appropriate for the two habitats. So the two species, when sympatric, evolved to be quite different in many additional traits. So if this evolution were to continue long enough, these species in sympatry could evolve into new species, as Darwin argued should happen in his Origin of Species. So here's a nice plot of large numbers of populations of limnetic populations in the red and the benthic populations in the green. And notice there's a difference in body size and gill rakers, and there's almost no overlap between the two. And whereas in solitary populations, they're all across the middle here. And we've got sex, the two sexes also. The large symbols are females and the small symbols are males. So the same pattern in both of the sexes in these species. Of course, although we predict that competition occurring over long periods of evolutionary time should result in divergence of the two populations into different portions of the shared resource, and therefore we should see character displays in these species, doesn't mean that anytime we see the two closely related species are different, it must be due to competition. So the reverse argument doesn't necessarily occur. So there's unlimited other reasons that could explain the difference. But if we do see the pattern of displacement in sympatry, but no displacement in allopatry, then we've got, like a natural experiment. So that's evidence in favor of competition as a driving force, at least competition in the past, the ghost of competition past, as Connell pointed out. So evolutionary dynamics predict that either species should diverge, character displacement, or maybe even speciation. Or in any case, there should be a selection towards reduction or elimination of the competition, but we could still have the ghost of competition past. Character displacement in sympatry, no displacement in allopatry. Okay, let's continue our consideration of species interactions and move on to the next type of interaction we want to consider is that's the consumer resource interaction. We're going to look at definitions, isocline theory, ecological and evolutionary outcomes of this interaction. The consumer resource interaction, by definition, is the interaction between two species in which one species' fitness increases as a result of the interaction for any reason, and the other species' fitness decreases for any reason as a result of the interaction. So, this, of course, occurs if the resource is a living organism that gets eaten by the consumer, but it can also occur with many other types of functional relationships, as we'll see. And the consumer resource interaction is also closely related to the competition interaction, obviously, as for example, when two consumers are dependent on the same limiting resource and as a result experience exploitative competition. There are several basic categories within this interaction type, and they have different terminologies. So, grazers are species who consume plant material as a resource and have no intimate relationship with the plant material they consume. So, they don't live inside the plant, don't use the plant as habitat, rather, they just find it and consume it. Grazers don't necessarily kill the entire plant they consume, although they certainly can, but they do kill a portion of the plant by necessity and therefore lower the plant's fitness. For example, sea urchins, which consume algae mainly, manatees, algae and many aquatic plants, um, herbivorous fish like Sarpasalpa here in the Mediterranean consume algae, seagrasses, and sometimes crustaceans. Chitons, like the lined chiton here, which is specialized to consume coralline algae. Parasites are species that consume any kind of organism but have an intimate relationship with their resource. Usually, if it live inside its tissues or on its skin, sometimes one species only, and sometimes many, depending on the life stage of the parasite, and don't necessarily kill its resource, often just make it sick, debilitate it. In the case of parasites, the resource they consume and live inside or on is called the host. So, bacteria and viruses, obviously, but also many animals can be parasites. Worms and crustaceans, especially in the marine environment, whales have species specific skin lice and barnacles. So, we've got some lice here and also barnacles on this a gray whale, I think. Isopod crustaceans can be parasitic inside the gill chambers of other crustaceans. Like there's a large female isopod inside the gill chamber of these shrimp, and a tiny little male, but the female is just gorged with eggs. Parasitoids are species that have an intimate relationship with their host, living inside its body, but by definition, they kill their host, like the alien in the movie Aliens, by consuming it from the inside out. So, a marine example is the ciliate Colinia, which is a paper by Gomez Gutierrez et al., which infects krill, lives inside the krill's body in its blood, and consumes the internal tissues of the krill until it dies and bursts, releasing large amounts of the ciliate. Not currently known how it infects the krill. So here's the final burst of the krill, the eggs released. So true predators don't live in or on the bodies of their resource, which is called the prey. Rather, they just consume it, and in the process of consuming, almost always kill it. Top predators in the marine environment are sharks and killer whales or orcas. All right, interestingly, some types of mimicry, namely Batesian mimicry, named after Bates, is probably the most common plus-minus interaction in which the plus species does not consume the minus species. Batesian mimicry is a particular kind of mimicry. So this is when one species mimics another in order to get an advantage. The species mimicked is called the model. In the case of Batesian, the model is toxic. Okay, so Scorpaena is a fish that's toxic or physically injurious when it's consumed, usually toxic. So if a predator or the mimic is fooled into thinking that the mimic, so here's the mimic, this is a Fowleria fish that looks like Scorpaena, mimics the appearance. So when the predator sees the Fowleria, and if that predator is fooled into thinking the mimic is actually the toxic model and then avoids it, well, the mimic obviously benefits. So obviously the Batesian mimic benefits from its resemblance, but what about the model? What about Scorpaena? What effect does the mimic have on Scorpaena? Does the model benefit or is it harmed by the mimicry? Or is there no effect on the model? Well, the model may actually be harmed by the mimicry. If the model is rare, then predators might not encounter it as often as they encounter the mimic. If they encounter the mimic often enough to learn that the mimic is perfectly fine to eat, they may also think the model is the same as the mimic, namely perfectly fine to eat, and try to consume it and kill or injure it. If the model is common, then perhaps predators don't encounter the mimic enough to learn that it is safe, and therefore mimicry does little or no harm to the model. Okay, so variable, but in general, you would think of this as a plus-minus interaction. Another kind of mimicry, which is Mullerian mimicry, is not a plus-minus, probably a plus-plus interaction, mutualism. So in Mullerian mimicry, many toxic species evolve to have similar coloration that signals toxicity no matter what species it occurs in. The color combination that almost universally signifies toxic danger in animals is the combination of yellow and black, like you see in bees and wasps. In most cases, animals with these colors are toxic, but there are also examples of Batesian mimics which use the same combination, although they are not toxic and perfectly edible, like syrphid flies, for example, have black and yellow stripes but are non-toxic, unlike the wasp models they mimic. So in Mullerian mimicry, potential predators learn to avoid the color combination regardless of the identity of the species they learn it from. And there's no fitness difference between the mimic and the model, so everyone benefits about equally. Okay, so much for the types of plus-minus interaction. So we have a lot of questions that are interesting for practical reasons, conservation and economic, and for basic reasons, understanding biological diversity, for example, and function. So commonly asked questions in research projects involving predators and prey: what kind of balance exists between predators and their prey? For example, why is the world green? Plant material is food for grazers. Why don't the grazers just consume all that green and convert the world to brown? What prevents consumers from driving their resource species to extinction? Should consumers evolve to cultivate their resource and safeguard it from extinction? Has this ever happened in nature? Is there such a thing as a benevolent predator? How can humans harvest living resources sustainably? What is sustainable harvest? What causes the outbreak of pests and diseases? Why don't their predators keep them rare? Can we control invasive pests by introducing predators that drive down their abundance? It's called biocontrol. What causes sudden disease outbreaks? How can we prevent and control these? How do predators and prey evolve in response to each other over millions of years? Or generally, consumers and resource, including parasites and host? So these are big questions, and many ecologists have spent their lifetime working on them, and they are still areas of active research. And we'll briefly sample them in the marine context in this unit of the course, namely populations, as well as the next, which is communities. And the study of population dynamics of predator and prey started with, guess who? Lotka and Volterra. They also had differential equations published in the same paper on competition, but these were predation models. So not only did they independently develop the equations for exploitation competition, they also independently developed equations for a simple predator-prey system in which the predator's fitness is determined only by the prey population and the prey's fitness is determined only by the predator. Okay, here we go. We consider the Lotka-Volterra model for the simple predator-prey interaction. So the conceptual framework is the same as before. We have two populations, this time a predator and prey. And within each population are organisms with the property of fitness, a property of organisms and not populations. And at the population level of organization, we have again density, growth rate, and dispersion. And as there's a feedback between properties of the population and fitness of organisms in both populations. Increase in density of the predator population causes a reduction in fitness of the prey. Increase in density of the prey population causes an increase in fitness of the predator. That's the plus and minus. So those are the two assumptions of the Lotka-Volterra equations for the predator-prey dynamic. Remember, in the competition equations, the effect on fitness was different. Increase in the density of either population caused a reduction in fitness of organisms in the other population. So, since for the purpose of explanation, we don't need to fully understand the details of the temporal dynamics, rather, we need just a semi quantitative picture. Instead of writing down the equations here, instead, you'll do this in the lab for this week. We just draw the isoclines on the phase diagram that relate N1 to N2 and try to think about how isocline geometry can answer the questions above. So, as far as the isoclines are concerned, a positive interaction gives a positive slope. Negative interaction gives a negative slope. Zero interaction gives zero slope. So obviously the positive slope is going to be the predator's isocline. The negative slope is going to be the prey's isocline. So let's draw the isoclines. So the prey population here on the x-axis, of course the x-axis has the carrying capacity for the prey. Let's say it's right here. So in the absence of the predator, the prey will increase to its carrying capacity. Okay, so if we start here and start adding predators, the carrying capacity is going to go down, 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 down as predators increase because the fitness of the prey is going down until we get to the shutout level of the predator right here. If the predator increases beyond this density, then the prey can no longer exist, and it's basically driven the prey to extinction. So this is the prey's zero growth isocline right here. So in the case of the predator, if we start at the maximum density in the absence of the prey, adding prey will increase the predator's maximum density. The isocline will have a positive slope, indicating that the interaction has a positive effect on the fitness of the predator. So we'll just assume that in the absence of prey is zero here, we'll just assume that the predator's fitness is also zero. So it cannot exist in the absence of its prey. So the predator itself goes to extinction. As a first assumption here, we'll change this. So the predator's isocline is positive. Adding prey increases the predator's maximum density. Positive slope indicating a plus effect on the predator. So in the case of the prey, we start at the maximum density in the absence of the predator. Adding the predator will reduce the maximum density of the prey, has a negative slope, indicating the interaction has a negative effect on the fitness of the prey. So we assume that the predator's density reaches a maximum, saturating curve here, because adding more prey may have no effect because the predator is saturated with prey. Now something else is limiting its growth, such as habitat or its own predators. Okay, so at a certain point, adding more prey doesn't really do anything to increase the fitness of the predator. It's reached its maximum. So here we should keep in mind there are several possible shapes for the predator isocline, and it helps to consider them and how they're determined by the rate of consumption of the prey. So we can think of this as sort of the maximum population density of the predator, a kind of carrying capacity again when we have abundant prey. All right, so what the predator isocline looks like depends on how it responds to prey density, how its consumption of the prey responds to prey density. And we can plot the rate of consumption of prey by the average predator individual, in other words, mass per unit time consumed, as a function of prey population density. So these curves are called predator functional responses. In other words, how does the predator function? In other words, speed of consumption of the prey. How does that function depend on the density of prey? Well, there are three curve types that have been defined and named in classic ecological literature. These are type 1, type 2, and type 3. Type 1 is just simply a straight line originating at 0. The predator consumption rate is unlimited and is simply a constant multiplied by the prey density. Functional response 2 is a saturating curve originating at 0 right here. The slope of this curve is highest at 0 and decreases to 0 at saturation. So the type 2 curve captures the realistic assumption that the predator individual reaches a maximum consumption rate because it has to capture and handle the prey before consuming it, and the limiting factor in capturing and handling and consuming is no longer prey abundance. It's just the mechanical ability of the predator. So it basically gets saturated by the prey, can't increase any faster. All right, functional response type 3, or the so-called hauling functional response, is a curve that is saturating at high prey density, but whose slope is not highest at 0 prey abundance, but rather close to 0 at 0 prey abundance. The slope of the curve actually starts at 0, then increases to a maximum somewhere in an intermediate value, and then eventually reaches a plateau. The curve is S-shaped or sigmoid shaped, so this is a logistic curve. And there are several possible reasons for the low consumption rate at low prey density. These could be, for example, low reinforcement of behavior. The predator has to learn how to find and capture the prey, and if the prey is not abundant enough, the predator does not get the behavioral reinforcement it needs in order to efficiently capture and handle the prey. So it's primed for a different prey species. But as the prey increases in abundance, the predator learns how to capture and handle that prey. Or another way to put it, if the prey becomes too rare, the predator switches to a different prey species. So this part of the curve can be due to predator switching or learning or reinforcement here. Regardless of the reason, if the prey gets sufficiently scarce, then the predator no longer consumes it. Or it also could be true that at low density, prey have some kind of refuge that prevents capture, and that refuge can accommodate only a limited number of prey. So as a prey increase in abundance, then they have to leave the refuge to feed and then become vulnerable to predation. So there are lots of reasons we could have this little curve down here, this sigmoid curve right here at the low abundance of prey. The importance of having the right functional response curve in your models, for example, hauling type 3, is illustrated by this Camphidol paper in which they show that these predators in the North Sea, the sea cod and North Sea whiting, actually positively switch to different prey species when one prey becomes rare and therefore have a type 3 functional response. So they found this by identifying their stomach contents, taking out their stomach contents and IDing it, and they found that prey that became less abundant completely disappear from their stomach contents. So that's a type 3, hauling type 3 functional response. So previous management models used a different functional response that predicted that they would drive their prey species extinct under certain management scenarios, which never happened. So when they did this study, they found that, okay, they had this type 3 response, which explains why they were not able to drive their prey extinct, because when their prey became rare, they switched to another prey. All right, so it's important to know the functional response of the predator in order to get the right shape of the predator isocline. For example, here down here, it might resemble more of a sigmoid curve at low prey density if there's a hauling type 3 functional response. All right, now a quick point I want to make here, thinking about these isocline analyses, especially the predator isocline, it makes you think of an important point here. The concept of carrying capacity, though sounding simple, is actually very complicated. So there are many ways to think about carrying capacity. First, you can define it as the maximum population density in the absence of any competitor or predators, where your consumption rate of resource balances the supply rate. Call that the resource K. Or you can think of it as the maximum population density in the presence of all of your competitors, which reduce your supply of resource and your carrying capacity. Or you can think of it as the maximum population density in the presence of all your competitors and predators, who reduce your fitness even further beyond the reduction you've already experiencing from your competitors. And that would be kind of an interaction K. So we can think of a resource consumption-based carrying capacity, and we can also think of an interaction-based carrying capacity. So when you use the term or encounter it in the literature, it's important to be aware of these distinctions and how, it's important to keep those things in mind in order to understand what we really mean by maximum population density. Okay, with that in mind, let's take a look at what happens with this phase space diagram. So if we start with these isoclines, saturated curve for the predator and straight line with a negative slope for the prey, how do the predator and prey populations grow or decline in response to each other? Can the predator cause extinction of the prey? So we'll add more realism to the isoclines as we consider them. We'll just do it graphically and not think about the equations for now. Okay, so first we see an important new result that we did not see in the Lotka-Volterra competition isoclines. In the predator-prey interaction, it's possible to get oscillating population chains, oscillating populations of predators and prey, which increase, then decrease, then increase out of phase with each other. So this oscillation phenomenon is always possible whenever you have a plus-minus interaction. Okay, see this oscillation potential? Let's zero in on where the two isoclines cross. So right here, where they cross, the predator isocline is almost vertical and the prey isocline is almost horizontal. So when the prey population is to the left of the predator isocline, the predator population decreases. When the predator population is above the prey isocline, the prey decreases. So if the two populations are in this upper left quadrant, both populations are decreasing. And we look at the direction of movement simply by adding the two vectors of population change of the two species to get the resultant vector of movement. So again, if they're in the upper left quadrant, both populations are decreasing, and they continue to decrease until they move into the lower left quadrant. Now the prey starts to increase while the predator continues to decrease. Eventually they move into the lower right quadrant. Now both are increasing. Eventually they move into the upper right quadrant. Now the prey decreases while the predator increases. Finally, they're back in the upper left quadrant where they started, but they're closer to the equilibrium point. So in other words, we see an oscillation in population densities of both predator and prey. So the four quadrants show upper left, both decrease. Lower left, the predator decreases and the prey increases. Lower right, both increase. Upper right, the prey increases, the predator increases, and the prey decreases. Okay? So for both populations, sometimes they increase and sometimes they decrease, and therefore, by definition, we have oscillations. However, notice we're not back in the same location where we started. We move closer to the point where the two lines cross, the equilibrium point. So if we continue very quickly, we end up at the equilibrium point where the lines cross, and the populations now no longer are increasing or decreasing. This, by definition, is a damped oscillation. The amplitude of the oscillation gradually gets smaller until there's no longer an oscillation. No matter where we start out on the isocline plot, we have a short oscillation. However, note that the equilibrium point is close to the y-axis. This means that it is possible, when the two populations are in the upper left quadrant, for the prey population to hit 0 before the predator population drops below the prey isocline and allows the prey population to start increasing. So like this. So in other words, anything causing the predator population to rise to high levels could result in extinction of the prey population. So this is a sort of isocline situation that's proposed to apply to the cod and whiting predators with many species of small prey fish. Yet the small prey fish populations seem quite healthy and at no risk of extinction. So what's wrong with the isoclines? Or more to the point, why haven't the cod and whiting driven their prey to extinction? Well, first, there are several ways to make the isoclines more realistic. For example, the prey isocline is probably incorrect. The prey isocline. Any particular prey species is not going to be at risk of extinction from the predator when it is at low density. This is because the cod and whiting generally switch to other prey species when any single prey species becomes less common. So this means that at low density, each prey species has a refuge. At low density, the risk of predation is much relaxed, allowing the prey population to increase when it falls below a threshold. How do we show this in the isoclines? Well, increasing prey population occurs below and to the left of the prey isocline. Therefore, to make the population increase at low density, we draw the isocline to the right and rising up at a threshold of low prey density. So this prey isocline prevents the prey population from dropping all the way to 0 as a result of predation. And we can also make the predator isocline more realistic by making it sigmoid or S-shaped, like the type 3 functional curve to indicate also that the predator shifts to a different prey species when the prey drops to low density. So here with the prey isocline moving up, the phase space movement is this direction, then this direction, then this direction, and eventually comes out to this equilibrium point. So the raised isocline prevents the prey population from going all the way to zero. And we could also show this predator isocline as a sigmoid shape like this instead of just a pure saturating curve. We'd have to redraw it to reflect the fact that they're not consuming the prey at these low densities. All right, one way to understand under what conditions oscillations occur and how extreme the oscillations are, extremity tending to result in exclusion of the prey, is to experiment with the geometry of the isoclines and see what kind of dynamics will result. So then we can infer what that geometry means in terms of how the populations are regulated in nature. So for example, what if the predator isocline saturates very rapidly at low prey density, very rapidly here, and crosses the prey isocline at a high prey density? Well, then there are almost no oscillations. The damping is almost immediate. So let's look at it. Here, the arrows almost point to the equilibrium point. So there's hardly any oscillation at all. And we go very rapidly to this equilibrium, which is far from the 0 point, which indicates a fairly stable system with unlikely outcome of extinction of either species. So we can plot the two densities right here. So the predator population just goes down and then stays stable. Prey population goes up and then reaches a stable endpoint. That's it. No oscillations. So what does this mean in terms of what's going on in nature? Well, if the predator isocline saturates very rapidly, as it does here, it saturates very rapidly. That means that something other than prey density is regulating the predator population. Something besides the prey density is preventing the predator population from rising to higher levels. So it's prevented from rising to higher levels, then that prevents it from overshooting the equilibrium point where the two isoclines cross. So it's not going to overshoot that equilibrium point. And if the predator population is prevented from overshooting the equilibrium point, then it can no longer oscillate. So if some factor other than prey population density, such as habitat limitation, scarcity of suitable habitat, or the predators of the predator itself, or predators keeping the predator down, something else like that is limiting the predator population, then oscillations are more damped than otherwise and may not exist at all. So we can draw some conclusions just by experimenting with the isocline geometry. If the predator isocline does not saturate quickly, we can get wide and predictable oscillations in both species. And this would occur if predators have a high consumption rate and little or no crowding or habitat limitation. Right? The result also is a high equilibrium predator density after the oscillations eventually stop and the two populations reach equilibrium. If the predator isocline does saturate quickly because of a low consumption rate, or crowding and habitat limitation, etc., then we have little or no oscillations and a lower equilibrium predator density. Okay, so we can find out a lot just by experimenting with the isoclines, and it makes a lot of sense. All right, so the next question is, how do we get permanent predator-prey oscillations? So we see those in nature. For example, the classic terrestrial example is the Canadian lynx and snowshoe hare populations across the Canadian boreal forest. So these populations have been in regular oscillation for 2 centuries now with no sign of any damping at all. How can this happen? Well, let's play around with the isoclines to see how this happens. Okay, so let's look at the isoclines again. Here we have a damped oscillation here when the isoclines cross at this angle. Here we've got no oscillation at all when the predator isocline is horizontal where it crosses. So how do we get permanent oscillations? Well, it depends on the angle, and the answer is simple. If the two isoclines cross in a way so that when the resultant vector crosses into the next quadrant, it is no nearer the equilibrium point than it was in the previous quadrant. Like this. When it crosses into the next quadrant, it's no nearer the equilibrium point than it was before. So that occurs when the prey isocline is horizontal and the predator isocline is vertical. So this happens only when the two isoclines are exactly at 45-degree angles from the vector arrow. 45-degree angle. If they're exactly 45 degrees from each other in each quadrant, then the head of the arrow is exactly at the tail of the previous arrow. So you have to come full circle to the same starting point. This is a permanent undamped oscillation. So this occurs only under one situation, when the predator isocline is exactly vertical and the prey's is exactly horizontal. So the exact opposite situation is when the predator isocline is horizontal and the prey isocline is vertical. Here we have no oscillations at all. That's this situation right here. This is because the vectors all point right at the equilibrium point. Okay, so the geometry of the crossing point is hugely significant. Right, what does this situation here mean functionally in the real world? It means that neither population has any effect on the other near the equilibrium. Whether the prey population increases or decreases just as depends on where the prey population is relative to this vertical isocline. So it depends only on the prey population. And whether the predator population increases or decreases depends only on the predator population. So there's no interaction. This is a case where there's no interaction between the two. They're just dependent on each other and they just reach each other's equilibrium point. I mean, they reach each's equilibrium point. So each population is limited only by itself. There's no interaction anymore. So under these conditions, the population acts like the other species is not present, rather like a single population under logistic growth with a smooth approach to carrying capacity, or more accurately, to its stable equilibrium population density. Okay, so we just, to go from one to the other, we just rotate the isoclines. So we start out with this one. You can rotate it down here so that the yellow is vertical and the red is horizontal. So we start out with this permanent oscillation situation, and then watch as we rotate. The arrows get closer and closer and closer to pointing right towards the equilibrium point. So this situation here is a permanent oscillation. And in this situation, whether a species increases or not depends only on the other species. So the interaction is maximal. The other species is the only determinant of whether either species increases or decreases. So there's no other limiting factor. They're just dependent on each other. We have a positive negative interaction between these two species, and that's the only interaction in the environment, the only important interaction in the environment. Okay, so if we move this more and more this direction, that means that the two species are having less and less impact on each other. And so there's no interaction at all between the two species, the predator and prey, anymore. And so they just increase to their carrying capacity smoothly, and that's it. So here's how we get undamped or permanent oscillations. The two species depend only on each other and nothing else. The predator consumes only the prey species, and the prey population is controlled only by the predator. That is, the prey never reaches its resource carrying capacity because the predator always keeps the population low. So this is a situation in which the predator isocline is vertical and the prey isocline is horizontal. And it turns out that this isocline geometry, which incidentally is the simplest relationship between predator and prey, was exactly the geometry proposed and analyzed in the original predator-prey differential equations by Lotka and Volterra, both independently. It's like they were reading each other's minds. Both of them independently came up with the same competition equations and the same predator-prey equations at about the same time. Lotka just basically a couple of years earlier. All right, so here's what the Lotka-Volterra isoclines look like. And so we've got the horizontal prey isocline. So we get zero growth of the prey no matter what the prey density is. Anything on this line is going to be zero growth. So the prey density has no effect on the growth rate of the prey, and the predator density has no effect on the growth rate of the predator. So the only influence of each species, the only influence that it has is on the other species. Okay, so presumably this isocline would eventually come down to zero at some point. All right, so this is a permanent oscillation. If you just do the plots of the abundance of the densities of the two species, you can see that it's just a permanent oscillation like this, with the predator just a little bit ahead of the prey. So the predator's trajectory is just a little bit ahead of the prey's trajectory. Okay. So this is the Lotka-Volterra predator-prey model right here. Many other isocline geometries have been explored since then, and by many population ecologists, including Nicholson-Bailey, the Nicholson-Bailey model for host parasitoid dynamics with subsequent refinements. So here we have no oscillations, and here we have stable undamped oscillations. And depending on the angle at which these cross, you can get in-between oscillations somewhere in between there. All right, so this is the Lotka-Volterra model, and then many others have proposed models where the isoclines cross at different angles, and different authors come up with different predator-prey dynamics based on these different isocline models. So, what we conclude from all this? Well, endless oscillations we predict should be found in simple situations and in communities with few species. So the predator and prey are affected only and primarily by each other. So the hare-lynx oscillation in the far north is consistent with this prediction. It's far north, so there are very few species there. So mutual sensitivity, we get oscillations, and if we have mutual insensitivity, then there's no interaction basically. So there's not going to be any interaction, any oscillations. Right, here's a marine example that apparently shows the same level of endless undamped oscillations, and this is the interaction between the sockeye salmon, which is the prey, and the predator trout, the rainbow trout of the Fraser River system of British Columbia, Canada. The salmon is an important sport and commercial fishery, and the population has shown wide oscillations throughout that time with a period of about 4 years. in this paper by Gwill and colleagues. So every 4 years, there's a large spike in abundance of sockeye salmon spawners. You just go out and count the spawners at the streams. So the last spike was in the year 2018, and there'll be another one in 2022. So every 4 years, the fishermen are lined up to catch the Fraser River sockeye salmon. And the difference in abundance between the peak year and the off years is about 100 to 1,000 fold difference. So this oscillation is very dramatic and something that the fishermen plan for every 4 years. What's causing the oscillation in the salmon? Well, one possibility is the isoclines cross in a pattern sort of like this, and that this is a classic Lotka-Volterra predator-prey oscillation caused simply by a strong dependency of the two populations on each other in the absence of any limiting factors on the populations outside the predator-prey interaction. So these authors wrote a couple differential equations to describe growth of the two populations. That's these here. All right, so here's the population growth rate. These are two logistic type curves with a few more parameters in them. So S is the prey and P is the predator. These are the equations for the population growth rate. So you set these to 0, and then you plot the two ends against each other, S and P against each other. So this would be P on the y-axis and S on the x-axis. And this plot here I just made myself. I put these equations into R and solved the differential equations in R. So these are different population trajectories of the two species using different parameters here. So this is a damped oscillation if there's some self-limitation in the parameters, and this is an undamped oscillation if there's less or no self-limitation in the parameters. So when they solved the equations numerically and plotted the results here, and I also did it myself, check, they found for reasonable parameter values, they were supported by field work. The equations give a nice oscillation in both species with a period of about 4 years. And this plot here is a phase space diagram again. So this is the isocline for the prey, and this is the isocline for the predator. And notice that this is vertical, and we've got even, it's not quite a horizontal one, but it's even, it's a positive slope, which means that prey is actually self promoting its population. Okay, so it makes the oscillations even more extreme. Okay, so we have the populations oscillating around like this, just like we drew the vectors before. So this is a permanent oscillation that never stops, but this is an unrealistic example. Okay, so there's got to be something more than this going on here, because it makes no sense that the predator should have a larger abundance than the prey. Okay, and also you have this population getting very close to 0 here, practically 0, which is also unrealistic without causing extinction. So there must be something else going on here that needs to be taken into account. So this is just a deterministic model. If you added random chance to it, then down in this region, this would definitely result in the extinction of the prey, because this is just right along the 0 point. Now, we still don't know if this is the reason for the oscillation. There could be other factors causing it, but it's hard to see what else could be causing such a predictable and long-term cyclic behavior other than something that is internal to the population dynamics of these two species. But, we can say that the model is reasonable and predicts the observed oscillation, so is well supported by the empirical facts. And you can see that the model and its predictions and compare them to the observed population oscillation, you know, um, in the data. So here's the prediction, and here's the data. Obviously, more things are going on, but at least we have the cyclic behavior predicted nicely with the 4-year period. Right, next question is, how do we get cases where there are wide oscillations, but neither the predator nor the prey go extinct? So this is occasionally observed, wide oscillations, but no extinction. Well, again, the isoclines can suggest reasons. If the prey have a refuge at low density, then that will prevent their extinction. So that is indicated by this isocline here, by the prey. And if the predator switches to a different prey when its main prey becomes less abundant, then that would prevent the predator's extinction when the prey falls to a very low value. So something like this. So a refuge at low density for the prey can be represented by a prey isocline that becomes vertical as the prey population density becomes small, and predator switching, the type 3 functional response, can be portrayed by a predator isocline that intercepts the y-axis well above zero. So in other words, when prey abundance is zero, the predator population can continue to exist and increase with substantial numbers with a low risk of extinction. Even with small prey values. And that's because the predator is not 100% dependent on this prey species, but rather can switch at low density, and this would show a type 3 functional response, as in the cod and the whiting. Switching. So we get oscillation that is permanent and undamped, but without any possibility of extinction in the two species. So this might be a more realistic example, a realistic set of a realistic model for the salmon and trout interaction. We get permanent oscillations with a stable system with no possibility of extinction on either side. And it might also apply to the cod and the whiting. These predators might have, both of them might have similar isocline geometry as what we're looking at here. So a little quick summary here of all that we've been looking at. So coexistence between predator and prey is favored by predator switching when the prey is scarce, refuges for prey at low density, strong self-limitation in both predator and prey. Oscillations are favored by little or no self-limitation, or the presence of self-promotion or self-acceleration. That can cause oscillations, cause wide oscillations. All right, so we've explained a lot about predator-prey interactions just by playing around with the isocline geometry. And that gives us some interesting insights into what's going on in predator-prey interactions. All right, that's it for today. Have a wonderful rest of your day. We'll see you next time.